Okay, yes, welcome everybody. Thank you for coming. So today is our CRM-Fields-PIMS lecture. This is kind of the main prize that the Fields Institute gives together with the other Canadian institutes, CRM and PIMS. So we are very happy and I'm very honored to introduce this year's prize winner, Professor Leah Edelstein-Keshet. So Professor Edelstein-Keshet's been is very decorated. I usually like short introductions. She graduated from Weizmann Institute in 1982, currently at the UBC, Emeritus Professor at UBC. She has many notable distinctions, including the John von Neumann Prize 2022, and she was most recently become a Fellow of Royal Society of Canada 2023. So without further ado, I welcome our speaker today.
Thank you very much. Thank you very much for the invitation. And thank you to PIMS, to CRM and Fields for honoring me with this prize, which is very humbling, especially when one looks at the list of people who've received it. So it's really an amazing and surprising thing. So before I get started, aside from welcoming all of you here, I want to particularly welcome two of the youngest members of the audience, Olivia and Anthony, and Maria, who've come here especially from Oshawa to hear this in person, where they could have done so by Zoom. So I'm going to tell you a little bit about some of my favorite topics in cellular math biology. But before I do that, I want to give you just a little bit of background of what is math biology and how did I get into it. And then we'll go on and talk a little bit about some of the problems in that field. So you can think of math biology in one of two ways. You can either think of it as an area of application that fosters and brings up new ideas that mathematicians can work on, and I'll give examples of that. You can also think of it as an area of mathematical tools and methods that can help to solve biological mysteries. So it can help biologists with the things that they want to understand. Okay, so how did I get into this particular field? All right, so my mother, this is a picture of my mother working in the lab. She was a marine botanist. And, looking through microscopes in her lab, I saw cells and all kinds of squishy things that were ultimately very fascinating for a little girl. And certainly I thought biology would be a great thing to get into. This is a picture of my father working. Yes, he was a mathematician, and mathematicians can work without microscopes. They can sit in their armchairs and think about math problems and prove theorems in their heads or perhaps with a piece of paper. No equipment necessary. But you can see that I had influences from both directions, and this is what eventually got my compromise to be in math biology. So where did all this take place? Actually, there's a lot of back and forth between two parts of the world. I was actually born in the Middle East. My parents moved to North America, eventually settled in Nova Scotia, where my father was at Dalhousie in the math department. My mom was at the National Research Council. And then I went back and did my PhD in Israel. And then I went back to Canada. So I'll tell you a little bit about my scientific trajectory and some of the problems that I learned along the way. And you'll see that I return to them years later in some of what I describe later on in my talk. So as a graduate student, my supervisor was Lee Siegel at the Weizmann Institute. And from him, I learned about self-organization and cell aggregation. So since we've got kids in the room, I thought it would be kind of nice to show a couple of movies that show how these cells that are basically like amoeba like to get together when they're starved. They eat bacteria, but if they don't have any food, they start streaming together into one site. They aggregate, they do all kinds of weird things, and eventually they form this multicellular, three-dimensional structure that creates spores that can then go on and find better places to live. So this is something I learned from Lee Siegel, and in fact, from him, I learned about the so-called Keller-Siegel model. So these are partial differential equations that describe the fact that cells move around randomly. They are attracted to a chemical that they secrete. That's a process called chemoattraction. And the chemical diffuses, it's secreted by cells, and it decays. So this was something that was sort of bread and butter, as it were, when I was studying with Lee Siegel. We'll come back to this a little bit later in the talk, and you'll see that this model has a generalization that we've been studying recently. From Lee Siegel, I also learned about reaction diffusion systems and pattern formation, in particular the Turing morphogenesis theory, the fact that if you have two chemicals that act as a local activator and an inhibitor that's more long-ranged, you can get patterns of these chemicals that are created spontaneously. So local and long-ranged, basically that means that they either diffuse slowly or fast so that they can influence things either locally or far away. And it turns out that these Turing so-called patterns have been invoked to try and explain many, many beautiful patterns that we see in the natural world. For example, patterns on fish, stripes and spots, or even this polar pattern on this goat, where half of it's on one color, black, the other half is in white. And we'll see that I'll come back to this particular polar pattern in a cellular context. Okay, a little bit later, when I was finishing off my graduate work and starting my first visiting assistant professorship, I went to conferences that were highly influential in New England. These were the Gordon Research Conferences, and they had them every 2 years, including one in theoretical biology and biomathematics. And the lucky thing there was I met Bard Umentrout, who became both a very good friend and a scientific collaborator. I'd say he's a real genius when it comes to hooking up models, understanding them, and so on. So with Bard, I got him interested in a problem that has to do with cellular pattern formation. And again, let's look at a movie real quick. So what we're seeing here is a kind of cell, a collection of cells called fibroblasts. And you see that they migrate, they stick to one another, and you can kind of see that they like to be in parallel with one another. And in fact, when they're grown in a very dense culture, they begin to line up in rows and form these parallel arrays. And so our question was, what is it that causes them to form these parallel aggregates? How can we explain that? So with Bard, we put together a model which had a little bit of a twist to it. On the one hand, it's a PDE model because it's got derivatives, it's got diffusions and so on, but it also has what you could call non-local terms. So what is this model about? We're trying to explain the directionality of the cells, and we're trying to account for the fact that the cells can line up and form dense aggregates in single directions. So let me explain a little bit about the ingredients that went into creating the model and where the equations come from. So in the equations, there are, we account for the fact that cells have an orientation, for the fact that cells can undergo random reorientation and realignment. They can contact other cells, and when they do so, they can decide to turn and stick. This can happen with individual cell contact or contact with a group of cells, and also they can leave the, this bound cluster. So each of these individual phenomena or attributes shows up in the equations in one or another of these terms. And some of these terms are non-local because for a cell in a given angle, cells in many other angles can come and contact and align with it. Now, these terms that you see here with the stars, they're actually what we call convolutions. So they're non-local terms that include an integral, something that sums up over many different neighboring cells. So here's an example of this term expanded out so you can see what it looks like. And the behavior of a model like this, it turns out, depends on the probability that cells will align when they come into contact. And that probability depends on angle because cells that are at 90 degrees might just simply walk over one another and not really stick. Cells that are at acute angle perhaps would have a higher probability of alignment. So this is an example of a so-called kernel, the function that sits inside here, where cells that are close to 0 degrees but no larger than, let's say, 50 degrees have some chance of sticking and aligning when they meet. And all of this, again, is dependent on the relative angle. So one of the things that we were asking is, if you initially start your cells off at all different angles so that the distribution is uniform, you can find as many cells in one direction as in another, what has to happen for this alignment to take place? And it turns out that this is really just simply a problem for which you can use so-called linear stability analysis. So what is that? So basically, you look at the uniform distribution of cells over angle, and you say, what happens when we add a small amount of noise, where that noise we'll assume has a certain wave number, a certain periodicity, and perhaps a certain growth rate, lambda. Will this noise actually grow and get amplified and form some kind of pattern or not? So a bunch of algebra later, it turns out that you get an inequality that has to be satisfied in order for this uniform state to get destabilized and pattern to emerge. So I'll just briefly show the kind of geometric flavor we get from analyzing this type of condition. But let me first point out that A, the parameter A here, is something that depends on the original properties of the cells. For example, it depends on M, which is the total density of the cells in all forms. It depends on the rate of binding and unbinding and so forth. And so if we assume that the turning probability looks kind of like this as a function of angle, then the inequality I just showed you reduces to figuring out when a certain parabola dips below a bumpy curve that is basically coming from this expression over here. So this is the parabola. Here we have an expression involving the Fourier transform of the so-called turning kernel. And so you can understand this process of instability by looking for a wave number, a value of k, such that this parabola shown with the dashed lines falls beneath this expression, which is the sequence of curves. So you can see also from the way that A depends on cell properties that when you decrease A, basically your parabola is going to flatten out, which will make it more likely that it'll be underneath this bumpy set of this mountain peak or bumpy set of mountains. And therefore, for example, if you increase the cell density, that will increase the chance of alignment. And of course, this sort of analysis is also complemented with simulations. Here I'm showing simulations dating back to the 1990s. So you can see that it was quite primitive, the kind of things that we could visualize back then. By comparison, nowadays, people can hook together much more interesting dynamic simulations of either bird flocks. Here I'm guiding them. There are many of these free on the web, so it's quite lovely to see what people have done more recently. Okay, so I got back to Canada after my PhD around and other things around 1989. Here's a picture of UBC, a little warmer in the picture than it is out here at U of T today, but of course we have our winters as well. And I started getting interested in the idea of how cells move around. So here is a classic movie, something like the Citizen Kane of cell biology, meaning a movie you watch over and over again because you know, it just got so much classic stuff in it. What we're seeing here is a white blood cell, actually taken from the blood of the chap who filmed this. And it's migrating between these little red blood cells. I'll show it one more time. And you'll see a little dot or a little squiggle here. It's actually chasing a bacterium. So white blood cells, also known as neutrophils, are part of our immune system. They're in our body to chase down pathogens and to allow us to fight infection. So it's very important for these cells to be able to move around and follow some kind of guidance cue to be able to find the sites of infection and do their thing. And you'll notice that as it moves, there's a front part, there's a back part. It's constantly deforming as it undergoes this, this motion. Now, what is it that allows it to migrate? What is it that powers the cell and pushes out the front? So it turned out, and this was known already for decades, that there is a structural, dynamically structural protein called actin that forms filaments whose tips are right at the front of the inside the cell, and they push this out, push the front edge outwards in order to allow that cell to move around. So back in the 1990s, when I started getting interested in it, people did a lot of work on trying to understand actin filaments and all the structures that they made. So in the lab of Gary Borsi, Tatiana Svetkina was looking at strands of actin versus these basket weave of actin filaments and all these distinct structures that actin could make. And so at UBC, when I joined with a PhD student, Gül Jivelakoglu, and another PhD student, Alex Mogilner, we actually tried to understand the alignment of actin structures. And you can see that in a way, we use the same type of mathematics. So having gotten familiar with alignment of cells and how that can happen, it didn't take too much additional thinking or twisting to come up with models that could describe the distinct structures that one sees in these cells in actin. So this, I really mean not to dwell on this in too detail, but just to indicate that you get a lot of mileage from the same kind of mathematics that you understand in one problem to apply to another. Okay, so what I've just described is really a bit of ancient history, and you could probably tell from this figure, this photo of me with Gül, that it was when I was back in my younger days. Meanwhile, the field moves on. So in the last 20 years or so, we're still, cell polarity and motility has really still been, I'd say, a hot topic biologically, and also from the point of view of trying to understand it with math and with simulations. So some of the questions that we've been asking is, well, first of all, how can a cell figure out my front should be here and my back should be here and I should go this way? And how does it move? What is the mechanism that allows it to move? And these are, I'd say, ongoing challenges, but I think some progress has been made by many groups, us too, but by many, many groups. Okay, so first let's think about the idea of polarity. So polarity means front versus back. So remember we had our little white blood cell, our neutrophil, there's a front and a back. How does it know which is which? So it turns out that aside from the actin in the cell, there are more chemicals that form a pattern that tells the actin where to go. So here I'm showing a pattern of a protein called RAC. The name doesn't really matter. All we need to know is that it somehow regulates actin. And it creates a pattern sort of in the style of what I told you about of the Turing reaction-diffusion thing, right? It diffuses, it interacts, and somehow by forming a front and a rear difference in activity, it can orient the cell. Now, actually, it's very complicated. So here's a figure that shows the internal workings of the cell computer. Think of this as the cellular microprocessor. It's not got silicon chips. Instead, it's got proteins that turn on and off like little switches and funnel the information down to the actin filaments. So we can think of this complicated network of proteins basically as signals that the cell receives, that tells it about its environment, a pattern forming module somewhere in the middle of which RAC is a member, and readouts, which includes the actin cytoskeleton. Okay, so it's very complicated, and we simplified it down to the bare basics. But the basics that we looked at still contain some biological facts that are really very, very important and allow it to explain how certain proteins can polarize, can have a different level of activity in a front and a back of a cell. So here, let's take this little icon and just simply look at it in more detail. And what we see here is that we have some protein that has two distinct forms. It can be active and do things and influence other proteins, or it can be silent, it can be inactive, and it keeps trading places. Active and inactive proteins keep switching, and they have different attributes. So the active RAC diffuses very slowly because it's bound to the membrane of the cell. This is an environment that's highly viscous. And so even though the protein is the same thing, it diffuses very slowly when it's in its active form. The inactive rack diffuses much faster and, as it were, can carry the message on a global scale. So we hooked together a PDE model to account for this very minimal example of pattern-forming cellular proteins. This is work by Yoichiro Mori, a former postdoc, and Sasha Zhilkin, a former PhD student, and it consists of reaction-diffusion equations for the active and inactive forms of the protein. And the key aspects of this model is the rates of diffusion are very different. The total amount is conserved. So mass conservation actually plays an important role in the behavior. And there's some kind of positive feedback so that when you get a little bit of the active protein, it will tend to make more, will activate more of itself. Now, the fact that the total is conserved really changes the character of the PDE. If we only had one equation, which was a reaction diffusion type, we would have traveling waves that would go through the domain. But the fact that we are conserving the total so that some of it is being depleted has an important influence. And here's an example of what happens. So what happens, let me play this several times. So first, the purple curve is the active protein. The green curve is the inactive protein. So please note that the inactive protein is almost uniform and decreases because it's being used up by the purple guy. Furthermore, please notice that a wave invades through the domain. In this case, it's a one-dimensional spatial domain, and it freezes somewhere in the middle. So even though the simulation continues, the wave freezes and forms a distinct difference between one end and the other, meaning it enables us to explain how these proteins form a kind of polar pattern. In this case, yes. In this particular case, we do. Later on, I'll show you some where we have periodic. This is a 1D with Neumann at the two ends. That's right. Okay, so now how do we put this into cell motion? So far, what we've been able to explain is the fact that there's a pattern that forms in the cell, and there's active rack on one end, inactive rack on the other. It turns out that the active rack will create the actin filaments. The actin filaments will in turn push the cell edge forward. And so we begin to get cells, cell shapes that can deform and migrate. So this model is actually slightly more complicated, but the basic ingredients are essentially similar. And it's a simulation due to Lisanne Renz, who's also a former postdoc. So the model I've just explained is kind of a very minimal model. And this slide is just to remind me to tell you that we've looked at many different realizations of the signaling that takes place inside the cell. I won't have time to describe all of them, but here's one other example where there are more ingredients, and you can see that this cell is even able not only to migrate, but to turn if you change one of the components and stimulate it. Recently, a talented student of mine was inspired by this classic cell biology model, a movie, to create a simulation that sort of mimics what we see in the classic movie. He's using here software that has become available in the last, less than 10 years, where now we can basically encode all of these moving cell simulations cheaply and freely without too many resources. So having this type of simulation also allowed us to attract some biologists and start some work with them. And that's what I'd like to tell you about next. So up until quite recently, all of the simulations that we've done, we had to basically make up parameters and try and make very rough estimates of many, many properties of cells in order to get things to work. Meanwhile, the biology has moved on so that now biologists can actually control the behavior of cells with light. So so-called optogenetic experiments use lasers to activate proteins like RAC in specific parts of the cell with high degree of accuracy. So for example, these are images of real cells, and here's a cell that's just migrating. This is from the work of Orion Weiner with Jason Town. And here you see what happens when they shine a laser on the front, on the side, or on the rear of that same cell. So basically what happens is the cell either moves and turns if you stimulate it with light on the side, or it reverses direction and makes a U-turn. And you can see the trajectories down below. Now, they have such amazing data, and they were able to supply data for cells that do not move, but only change their behavior over time, change the level of the proteins inside them over time. And with that, essentially, we were able to take the same model that I've told you about, which we nicknamed the wave pinning model, add minor ingredients that they felt were essential to explain what they saw, and basically fit parameters to their data. So in other words, actually fit our models of several sorts to their data and get behavior that was very similar to what they saw. So for example, when they stimulate the cells first with a local light source on one side, and then switch it to globally illuminating the whole cell, they find that the cells by and large make these strange turns from one direction, reversing their direction from clockwise to counterclockwise. And it turns out that the wave pinning model on its own couldn't do it. When we add the inhibitor that they suggested, it didn't quite work. But when we added the inhibitor plus one other ingredient that's known to be there, then we could account for the behavior. And now all of this is happening with parameter values that for the first time actually are fitted to experimental data. So I got really excited by the fact that we've been playing around with these models for years and years, but this is the final time when we can actually get parameters for real cells. At the same time, as we're doing these collaborations with experimental biologists, I also have a student who is working on the analysis of systems like this. So in this particular case, not only does RAC create the spontaneously initiate the filamentous actin, the actin also acts as an inhibitor. And so putting that together, work by Jack Hughes has led to some really interesting analysis that we did joint with a mathematician, Arik Yocheles. So this is full PDE bifurcation analysis where Jack was able to find regimes where there's coexistence between polar and traveling wave solutions. So you could get a cell. Here we show a bifurcation diagram where on this axis we've got something that relates to the fact that actin inhibits the activity of RAC. And we see that on the diagram, we see curves for the uniform states from which polar solutions emerge and from which traveling waves emerge. And it turns out that there's a region over which they can coexist. What does that mean? Basically, it means that you can say that a single cell that has one set of parameters, the same mechanisms, everything's identical except for initial conditions, that's enough to give you one of two very distinct behaviors. That cell might be polar. In other words, it might have a distribution of the actin that is high at the front and low at the rear, or it might have waves of actin that travel around the rim of the cell. And so now we're looking at periodic boundaries, and we're looking at these PDEs on the edge of the cell and showing that the cell can either be polar, it can have a pair of waves moving around, it can have a single wave, in which case it will make a big turn, or sometimes multiple waves, and in that case, you see a kind of ruffling of the cell edge. So in a way, you could say that we've come back to this single cell motility, both in understanding the way that the internal mechanism works and also the shape that's created as a whole. Okay, so in the last part of my talk, I want to go up to the next level of organization and talk about what cells do when they get together and how do they coordinate to form a whole. So just quickly, there are many topics that we worked on in collective behavior of many cells, and I don't have time to go through them in detail, so I'll just give a taste of one of them before I go on to give you more detail on a slightly more mathematical version that we worked on recently. So just quickly, it's known that cells can collectively invade into places where they should not. So for example, in diseases like cancer, there can be groups of cells that break off of a tumor and go elsewhere. And we worked on data that was created by Andrei Levchenko and Jin-Suk Park in Yale. And my master's student, Naba, at the time, created an ODE model for the internal regulation of the cell. And basically, it was a circuit that either could emphasize cell-cell adhesion and have the cells holding hands and maintaining cohesion, or depending on this central protein, it could go to the opposite extreme where the cells let go and start to move. And here comes RAC, the same protein that promotes that motility. So that's one example of a project we worked on. But the one I want to spend more time on is a recent work that we've done on cell cluster formation and persistence. And this is a project that was motivated by Paul Kulisa, who is a biologist who originally was an aerospace engineer. He decided that biology is just more fascinating and has more interesting challenges than rocket science. So he went into biology. He grows embryos of chicks in eggs, and he can actually watch and see how they grow. And he is interested in the formation of various structures within the embryo. And one of those structures, the nervous sympathetic nervous system, in its formation, there's a cluster of cells that somehow has to migrate over distance and maintain a group of cells together that eventually connect up to other neurons. So that's what motivated our work. This is just a quick little example of a group of cells, simulation. They're talking to each other by secreting chemicals that say, hey, come this way, come close to me. And also they're talking to each other by saying, don't get too close. So they are secreting both an attractant and a repellent, and somehow that enables them to do their thing. But what I want to describe is slightly more mathematical flavor of work we've done recently. And it was inspired by this very nice paper that came from the group of Jose Carrillo with Falcao and Ruth Baker. And in that paper, what they were talking about is a density of cells that changes as a result of motion of the cells, where the motion is in turn described by the gradient of some potential that the cells themselves create. So this is in a very low Reynolds number regime, and so the velocity is proportional to the force being the gradient of some potential. Now, you'll notice there's this star appearing here. As we saw before, this is a non-local problem because somehow the potential that the cells are creating influences neighbors, not just locally, but has some kind of slightly more long-range influence. Also in their paper was something interesting, that these non-local kernels and these convolutions can also, under certain circumstances, be approximated by retaining higher order derivatives with certain coefficients that are moments of the kernel. So that's what was in their paper, and our contribution was the following. First, we chose a particular kind of potential, so-called Morse potential, which really just means that the spatial description of it is exponential. Next, we showed that this Morse potential actually links to an underlying generalization to the Keller-Siegel model. I'll say more about this later. And finally, we classified what the conclusions would be, what would come out of it. So here is a model that really looks very similar to what we saw earlier in the Keller-Siegel model, except instead of just one chemical, it's got two. It's got an attractant and a repellent. Now, just to remind everyone, what is chemotaxis, especially the young members of the audience here. What's happening here is you've got cells, and there's a pipette releasing an attractant. So these are dictyostelium cells, they're amoebae. They love cyclic AMP. They're going to migrate towards sites of cyclic AMP. And this is a pipette that releases that attractant. And chemotaxis is the process whereby the cells are being attracted to that gradient. Okay, so that's what these equations are basically describing. And it's kind of nifty that, just to go back a bit, because these chemicals diffuse much faster than the motion of the cells, we can do a separation of time scales where these two equations are basically put on what's called quasi-steady state, as shown up here. We've got boundary conditions where at the origin there's a cell secreting chemical, and you can show that solving these equations by the Green's function method gives you exactly what we said. It gives you a set of exponentials. So the Morse potential comes up very naturally. And here's an example of the profile you would get if you put one cell at the origin and it's secreting its chemical, then essentially the chemical profile around it, and therefore the potential would look sort of like this. Okay, so as I mentioned, we started off with a set of 3 PDEs, and by virtue of this kind of reduction, we get a single non-local equation. And the potential now depends on these two quantities, the repellent and the attractant. Moreover, we can, through our analysis, we can say exactly what are the spatial ranges and the magnitudes of the potential in relationship to the original parameters of the model, the diffusion rates, the rates of secretion, and so on. So the repellent, if we had a cell right here at the origin, then around it there would be a kind of tent shape of repelling chemical. Don't get too close to me, it says, and another potential due to the attraction, the attractant, and altogether it would endow the neighborhood of the cell with a kind of potential almost looking like a little Mexican hat, where neighbors would want to get close but not too close. Okay, so we simplified further by ignoring random motion of the cells. We assumed that the potential was not too long ranged, so that it's okay to approximate the problem with higher derivatives instead of the integrals. And that allowed us to explicitly solve for the shape of the cluster, the density of the cells. And here it is. The problem becomes basically linear, higher order ODE. You've got solutions that are basically cosines, trig functions, and moreover, you can relate everything down to the attributes of these kernels, the attraction, repulsion, magnitudes, and ranges. So that means that we can also figure out a classification of everything we think is going to happen. And when we do so, and here we do it in terms of dimensionless quantities, ratios of the magnitudes and of the spatial ranges, then we find that in order to get a robust, nice cluster, we have to satisfy certain conditions. We have to be in this part of the parameter plane, and here simulations are also showing that in fact you get clusters that form over space and time only in this region. The other regions, the cells disperse, and in some cases they aggregate so much that they collapse to a point. Finally, we can take all that analysis and reinterpret it in terms of the actual biology. It gives rise to an inequality that tells us that in order to have a robust cluster, the ratio, we're seeing here ratios of the repellent to the attractant attributes. So somehow the repellent sensitivity and rate of secretion, its value relative to the same thing for the attractant shouldn't be too low, shouldn't be too high. It should sort of be balanced between these other quantities. This is the ratio of the diffusions. This is the ratio of the decay rates of the chemicals. And if you're in that range, you can form a cluster that will be robustly maintained. simulations also can show the same kind of behavior. And that's basically it. So I'm basically at the end of my talk, but I want to just briefly summarize and kind of indicate some of the themes that I've tried to describe. First of all, in the continuum models, I've shown you both local PDE, reaction diffusion examples, and also non-local integral PDEs, where the non-local term was either in the source term, where there was instantaneous transitions, in the case of aligning cells, or possibly in the velocity term due to non-local potentials. I showed a couple of examples of discrete models. I didn't spend too much time on that. Also, another theme was that of self-organization. On the one hand, reaction diffusion models, as in the Turing mechanisms, produce pattern either if there's a local activator coupled to a global inhibitor, or in the case of wave pinning, some kind of local activation coupled with global depletion of something that gets used up. But also another way to self-organize, in the case of cells, is this chemotaxis model where you're repelling cells locally so they don't get too close to you, don't pile up in one tight cluster, but at the same time you're attracting them to come closer if they're far away. So for all of you, both mathematicians, biologists, and aspiring scientists, there's a lot of amazing phenomena out there in biology. And I've just scratched the surface here, so I hope this motivates, especially the young people, to go into some of these beautiful and very interesting fields. So again, I want to thank Fields, CRM, and PIMS for the great honor. To UBC Mathematics, they've been fantastic colleagues to me for over 30 years, to NSERC for funding, and to all of the young scientists who have been part of this journey. Thank you very much.